Good afternoon, everyone. So the title of our paper is the functional neuroimaging of self-referential encoding with age. So let's get started. This is the table of content. But before we really talk about the paper, we need to have a quick recap of what is self-referential and the concept related. First of all, what is self-referential processing? According to the levels of processing model of memory in 1972, it said that the depth of the processing affects the storage of information. In other words, the more meaningful the message is, the higher chances we are going to remember it. And they find out that the information that is related to self is being memorized the most, and that is the self-referential effect. So an experiment in detecting the brain region while having the self-referential effect is being set up, and the task of it is the participant will review and judge the adjectives given describing the personality traits, and there are three conditions which are the words in relation to another person, the printed format, and to the self. And the results shown that the participant remember the best in the condition that words are related to self, and the fMRI shown that MPFC was strongly activated when in the self condition, but not in the other two. So let's talk about the paper. Our paper topic is how aging affecting the memory formation. So there are differences between the old and young generation in the aspects of the activities of the brain region for encoding memory. And from the research perspective, the past research focuses on the successful encoding is also different. Speaking of the brain region for older people, the activation of medial temporal junctions and the parietal cortex decreased. Therefore, they needed additional regions, for example, prefrontal cortex for encoding memory. For the young adult, the initial encoding of information into memory activates a network of regions, including inferior frontal and medial temporal junctions. Regarding past research focuses of the successful encoding for the old adult, many studies have been focuses on assessing the encoding of relatively neuron verbal and visual, and for the young adult, they assess successful encoding or reviewing whether the brain region shown increase for encoding the specific type of information. For example, emotional information and the region amygdala is related. And in the middle of the Venn diagram, it tells us the formation of the emotional memory is the same between two groups as they both involved in the brain region amygdala, fusiform junctions, and the orbital frontal cortex. Thus, it comes to our gap that there has no study examining whether aging will affect the neural process of the cell referential memory formation. So, what does the previous study tell us about the relationship between aging and self-referential? As mentioned, NPFC activates when self-reference information is successfully encoded. However, it also connects to the other region, which is the parohippocampus junctions and the anterior prefrontal cortex, and it also demonstrated that both young and old group will activate NPFC when thinking about oneself. However, that is not enough. What we want to know is the extent to which aging is affecting the NPFC and other regions. Thus, it comes to the aim of this research. We would like to investigate whether aging is associated with the organization memory networking of self-referential information.
To study the specificities of neural responses, previous researchers test the extent to which aging reduced the specificities of neural responses through self-reference experiments. The experimental results show that in the case of passively viewing pictures of a single object, the ventral visual cortex of young people, including the fusiform facial areas and the parahippocampal place areas, show specific neural responsive to certain stimuli, whilst the elderly show less category selectivity. When watching a complex scene composed of focused objects with a meaningful background, older people show weaker neural responsive to the object selection areas in the brains than young people. And there is no difference between the two age groups for the background selection area. Moreover, another studies found that with age, the bilateral natures of the brain increase, leading to a loss of specificity. Research showed that young people only need one hemisphere to complete the task. Among them, the left prefrontal cortex is involved in verbal materials, and the right one is involved in the processing of spatial materials. The elderly needs to use two hemispheres to complete the task, and both hemispheres are involved in the processing of two materials. It can therefore be concluded that the specific decrease in the ventral visual cortex seems to reflect the loss of functions with age. This decrease in the hemisphere specificity was explained as a compensatory function. That is, the elderly need to use additional resources to perform challenging cognitive tasks. So far, the research on the age-related change in the neural response to the passive viewing of specific type of stimuli is mainly limited to the visual domain, while the bilateral age difference is identified primarily in the difficult cognitive task. However, according to previous research findings, social information represents another highly specialized domain for young adults, particularly in the neural response to thinking about themselves and others. Among them, the left inferior frontal cortex is more involved in the judgment of others, while for the elderly and young people, the medial prefrontal cortex is involved in self-judgment. From this, it can be inferred that the specificity of the neural response to self-related information is preserved with age. But these studies cannot strongly explain the age difference in MPFC recruitment because these studies is compared in a relatively simple objective judgment task. The specificities of the elderly neural responses to self-referencing in objective judgment tasks may collapse under conditions that require more resources. And previous researchers believe that encoding draw on more resources demanding processes than our previous comparisons between self and other referencing. Therefore, these studies will serve as a further test of the specificities of the response of MPFC with age.
Now, let's move on to the method part. First, let me introduce you about the design of this study. This study used a subsequent memory approach by Brewer and Wagner, and analysis to identify brain activity during episodic memory encoding. This approach aims to investigate the effects of aging on the activity of brain regions, which is associated with memory foundation and domain specificity of neural responses. As this study mainly focused on the effect of aging, young and old adults' response on the adjectives would be compared. Also, response on 3 different conditions would be compared, which the conditions would be explained later. Details on this will be addressed in the next slide. This research recruits 17 young adults from age 18 to 28 and 15 older adults from age 61 to 80. Now let's take a look at the procedures of this study. There will be a total of 144 adjectives for participants to encode, where participants would be responding to three orienting conditions, self, other, and case about the adjective by a yes/no button. For example, for self condition, participants would be responding to questions like, does this adjective describe me? For other condition, participants would be responding to questions like, does this adjective describe Albert Einstein? For case condition, participants would be responding to questions like, is this adjective presented in uppercase? The other condition works to provide a socially meaning comparison for the self condition, where it would be likely that distinct brain regions would be active in self and other condition, while the case condition works to provide a non-personally meaningful perceptual judgment. The adjective will be assigned to each condition balancedly, which means that 48 adjectives for each condition. E-prime software would be used to rotate the assignment of adjectives to each condition. Also, a point to note is that participants were instructed to respond to each adjective quickly. In the start of the study, participants first encode 48 adjectives in each condition, which they respond yes or no for each condition's question. After 15 minutes of delay, they will receive a recognition test, which have not been informed to the participants beforehand. Participants need to decide whether or not they had encoded each word previously in the study, and also respond by the yes/no button. This is done to test participants' memory after 15 minutes and see which kind of adjective do they recall most. After this, young adults' response on each of the conditions will be compared with old adults' response on each of the conditions. For example, both young and old adult self-condition will be compared, so as the other two conditions. Also, the response in each of the conditions by young and old adults would be compared. For example, response on self-condition will be compared with case and other condition in the same young participant, so as other old participants.
For neuropsychological and demographic measures, there are 5 items that are measured in this study, and they are education level, health, vocabulary, digital comparison, and mini mental state examination. And the results tell us they are all high-functioning adults with similar educational levels, with wide range of vocabulary, a good health and well-being. The young adult group has a relatively faster processing speed than that of the older adult group. And for image acquisitions, by Siemens Allegra 3D scanner with EPI sequence, and also a high-resolution anatomical image with magnetization prepared rapid gradient echo sequence. And there are 5 actions for pre-processing. And the researcher tried to correct the slide time. And then we realized for correcting motions and normalize to Montreal Neurological Institute template. And also to resample to 2 mm cubic fossil and adjust spatial moving to a 6 mm full with half maximum atrophic Gaussian kernel. And SPM2 software was used for pre-processing and data analysis. And for the data analysis, the subsequent memory events related designs was used in this study, and including the 6 regressions, and they are self-remembered, self-forgotten, other remembered, other forgotten, case remembered, and case forgotten. And the yes responses are recognized as remembered, and then while the no responses are recognized as forgotten. The researcher tried to explain it with HRF. And the researcher contrasts the images in self and other persons' conditions by this formula. the net self-remembered responses minus the net other remembered responses. The researcher compared the contrast for young and older adults in a random effect group analysis and two sample t-tests. And he also used the MassBar software to extract the percent change of the signal from the region he interested to obtain the common region of activity for the both group. He created a mass of fossils for the first group and figure out the overlapping regions activated in the second group. And using the Fisher methods in conjoint probability of p smaller than 0.001. So let's pass the time to the next groupmate to talk about the results of this study.
Now let's take a look on the behavioral recognition performance. There's the main effect of the conditions, which is two levels, self versus other and others versus case. The recognition accuracy is the self is highest than the other and then the case. There's also main effect of the age. The recognition accuracy shows that younger people has higher performance than the older adults. But in terms of the interaction effect, there's no interaction effect at all in here. The research results are consistent with the self-reference experiment conducted by the previous research studies.
fMRI have been used to study the reactions of different brain regions. Here, I would first introduce some of the brain regions that has been selected to test for reactions in different conditions to give a brief understanding on those brain regions. MPFC is a brain region respond to self-referential, which means self or others condition. PCC would also respond to self-referential, memory-related processes, and subsequent forgetting effects. ACC will react to self and other conditions, age-related differences in its response to conflict and under challenging memory condition. In the previous study and analysis common activity in young and old adults, no regions were significant, which contrasts to the result of this study. A number of regions emerge as exhibiting age difference in the interaction of self or other interacts with remembered or forgotten adjective. For all of the selected regions, the graph suggests that age difference in self-referencing during encoding results from reversals in the pattern of activity across the two age groups. From the image of the young adult group in the left picture, it can seem that the regions of A, which is ACC, B, which is MPFC, C, which is PCC, and D, which is inferior PCC, indicate the subsequent forgetting effects. Research shows that the activity level of the regions for subsequently self-forgotten items was higher than subsequently self-remembered regions. The response in ACC and MPFC of young adults group shows an opposite pattern mentioned above, which indicates that the activity level of the regions for subsequently other remembered are higher than subsequently other forgotten items. Comparing with the young adult group, the older adult group shows that the activity level of the region for subsequently self-remembered was higher than subsequently self-forgotten items. Also, the activity level of the region for subsequently other remembered are lower than subsequently others forgotten items.
In the experiment, there is no activation in the condition of self or other remembered or forgetting across age groups. This is pretty consistent with the previous research, which the VMPFC can be activated for subsequent recognized items compare self and other judgment piece in PMPFC. The result is very consistent in terms of the neural activity. The self-remember is score higher than the self-forgotten, and other remember are pretty similar to the other forgotten. In terms of the age difference, the elderly score better to identify between self-remembered and self-forgotten items.
After the sharing of the results section, now I'm going to reflect on the limitation and future direction of this study. One of the limitations of this study is low statistical power. It refers to imbalanced sample. The size of samples in one group differs from another group. Low statistical power will increase the possibility that a statistically significant finding represents a false positive result. For example, in the self condition, participants perform quite well in it, and insufficient data could be obtained in the forgotten past. Therefore, there is only weak evidence suggesting whether young adults and old adults use different brain regions during successful encoding of self-referential information. Another limitation would be limited number of trials. Some questions of interest were unable to be addressed due to the limited number of trials. In terms of elaborative encoding, this study failed to examine whether descriptors of oneself could differ from those that do not due to age difference. Also, this study failed to compare the behavioral frequency between yes and no responses during self-referencing for both young and old adults. To improve the validity of the study, the researchers suggested to include multiple intermixed trial types and balance the sample size between young and old adults group to examine the unexplored research interests, such as whether age differences exist in memory formation for referencing the self or others. The neural involvement is different between different age groups during self-referential coding, and the behavioral frequency between yes and no responses during self-referencing.
The researchers can examine whether the medial prefrontal cortex response to self is selectively enhanced in comparison to others by increasing the statistical power, such as recruiting more samples. They can also examine whether age differences present while subjects successfully encode self-related information, which can further explain age difference exists in subsequent forgetting effects. Next, we'll move to conclusion. The authors concluded aging changes neural activity that is associated with memory formations of self-reference information. You can see that there are age differences in brain regions that are related to encoding of self-reference information. In self-referential processing, aging affects the region that is responsible for modulating memory formation. In prefrontal and parietal regions, young adults show subsequent forgetting effects, while older adults show subsequent memory effects. It reflects that the difference between young adults and older adults in elaborative encoding process to encode information about self and others. After studying this paper, we have a more concrete concept of self. The concept of self is a system composing all the information and is a strong and clear sense that we are the personal agencies of our own, including the memory. We own the authority of ourselves and know ourselves the best, as we are born to have the ability to recognize ourselves, even though one day we are getting old.
This paper also reveals some concepts learned during this course, such as self-referential effect. Medial prefrontal cortex activity is associated with self-referential processing when compared to processing words in relation to another person or the printed format of the words, according to a study. You use a different source of information when determining whether a trial is self-descriptive as when you decide if an individual has the trial. In other words, people are good at remembering trials related to themselves as well as judging whether they are self-descriptive or not. Our self-perceptions rather than events in our lives are what we use to determine whether an adjective is self-descriptive. However, when judging others, we often focus on specific instances when the person may have demonstrated behavior associated with the adjective.
We should note that one's memories cannot be defined as good or bad by looking on just one aspect. This is the end of the presentation. Thank you. And here's the reference.